Olfaction, we'll start with that. What's olfaction again?
Smell.
Sense of smell. And then we have gustation, what's that? Taste. And then we have vision, which we all know what vision is. So olfaction and gustation, they both are very similar. They both involve chemoreceptors. In the case of olfaction, you're detecting chemicals that are dissolved in the air, versus with gustation, you're detecting chemicals that have dissolved in your saliva from the foods that you eat. But they both involve chemoreceptors. They send signals, obviously to the cerebral cortex. There's a primary olfactory area on the cerebral cortex that makes you aware of smells. There's a primary gustatory area that makes you aware of tastes. They also both have a strong link to your emotional brain, your limbic system, and that's why certain smells and tastes can elicit a strong emotional response. And then also too, you probably know this, they work together to give you kind of the full food experience when you're eating. I guess if you were a chef, you'd probably, anyone in here a chef or anything? I had a chef one time. What was that? I'm not, I can barely make a peanut butter and jelly sandwich. Anyway, gustation, olfaction, they might argue that vision is also like, you taste with your eyes, whatever. But gustation, olfaction, they definitely work together when you're experiencing food. And you've probably noticed that when you have a cold and you've knocked out olfaction, the food doesn't typically taste as good. So there is kind of that interplay. Now in terms of the anatomy for smell, let's cover that first. The anatomy for smell involves something in the roof of your nose called the olfactory epithelium. So inside your nasal cavity, at the roof of it, is this olfactory epithelium, which they've kind of enlarged here so you can see it. It's just like any other epithelial tissue, but in the tissue, there are three important types of cells. The first cell is arguably the most important because that's the actual smell receptor. So the olfactory receptors, they also sometimes refer to them as olfactory sensory neurons. It's the same thing. I don't really care which name you use, like on a practical. So these are the guys that actually detect the smell. They're the guys that on their if you looked on the cell membrane of the olfactory receptors, they would have little chemoreceptors that are able to attach to chemicals that are in the air that you breathe. And then, that would trigger a graded potential, et cetera, and then you perceive it as some kind of a smell. Your nose literally has somewhere around 10 to 100 million of these olfactory receptors. Any chemical that has an odor to it that your receptors can respond to, they refer to as an odorant, right? Obviously some chemicals don't have a scent to them, so those wouldn't be odorants, but any chemical that has a smell to it that they can respond to is called an odorant. There's two other types of cells that aren't as famous, but they're still important that are in there, something called supporting cells and basal cells. So you can see all three of them then in this picture. So this is a super duper close-up then of this little area here. Notice you got the olfactory epithelium here. Underneath it, you have connective tissue. Then you have the bone, the cribriform plate of your ethmoid bone. And it had these little holes in it that allow kind of the neurons, the axons to kind of go up through there, and then synapse, and then it becomes part of, cranial nerve number 1, the olfactory nerve. One of the things you're going to have to do on Thursday is you're going to have to look at microscope slides of the olfactory epithelium and be able to tell these three cell types apart on the microscope slide. So if you're looking here, this is the olfactory epithelium here. This little area at the top would be the connective tissue. And honestly, you're probably thinking, if you're like me when I first looked at this, how the hell am I going to tell these all apart? What you can see is a bunch of nuclei, but how can you tell, whether you're looking at a supporting cell or an olfactory receptor or a basal cell? And the answer is just kind of based on the relative position of their nuclei, all right? If you have a nucleus and it's kind of located down towards the inside part of your nose, in other words, down in this area, chances are that's going to be a supporting cell, because supporting cells, that's where their nuclei are located, down kind of close to the inside part of your nose. Versus if you have a nuclei that's kind of close to where the connective tissue layer is up here, in other words, these nuclei, those are going to represent the nuclei of basal cells, because that's where they're located. And then if you have a nuclei that's somewhere kind of in the middle, then that would be a nuclei of one of these olfactory receptor cells, and you can see them here. Notice the olfactory receptor cells are actually bipolar neurons. Remember from chapter 12, you had multipolar neurons, bipolar neurons, and unipolar neurons. Bipolar neurons are the least common neurons that you have, but this is one of the places where you see them. Right? So, on a lab practical or something, if I were to point and say, like, what is this cell down here? You should probably say supporting cell. If I pointed to something that's up here closer to the connective tissue, then said, what cell is this? You should say basal cell. And if I point somewhere in the middle, just say like olfactory receptor.
So
okay.
the top, the nose part will be the bottom part. Like
the inside, yeah. I mean, the inside of the nose would be down here. So, like down here would be the inside of the nose.
Okay.
Yeah. All right, this is where I want to skip this part and go to gustation then. So gustation is simpler than olfaction. There's only 5 primary tastes. Did everybody learn about these 5 primary tastes like in kindergarten or something? I feel really old because they only told me when I was back in kindergarten, they only had those first 4. I never learned umami.
Yeah.
Anyway, I don't know. Who learned umami in school? That probably means you're young. All right. Anyway, whatever. Okay, it's like a Gen Z thing or something. Umami, fine. We got umami. It's meaty. It's supposed to be described as a meaty or a savory taste, I guess. I don't know. But anyway, those are the 5 primary tastes. Now, when you eat a food, it's a combination of those sensations coming in from the chemicals that are in the food, plus the olfactory sensations that are coming from the scent of the food. And then also, don't forget, it's also the tactile sensations when you eat. Like, is the food hard or soft? Does it like crunch? You have to chew it, right, and it actually crunches. Is it hot versus cold? So that's also a part of it. Now, those type of sensations, they're not carried by chemoreceptors. Those are going to be handled by your standard sensory receptors like you have in your skin, except you have them inside your mouth too. So like Meissner's corpuscles, Pacini, those type of touch receptors, they're also inside your tongue, and they're telling you, is this food hard? Is it soft? You also have thermoreceptors. Is the food hot or cold? I think I mentioned this before, but if you're a type of person that likes spicy food, what does spicy food actually stimulate?
Pain.
Yeah, pain receptors, right? So nociceptors inside of your mouth are responding to that. In terms of the anatomy, if you look at your tongue in the mirror after class today, or whenever you look next, you're going to notice all these bumps on it, right? And before you took this class, maybe you thought those bumps you were looking at were the taste buds, okay? That's what I thought before I became not stupid. Yeah. And just joke. No, seriously, I used to think, those are taste buds. They're not taste buds. Those big bumps that you see, those are called papillae. Okay? If you could get out a microscope and look at those papillae, then you would see the little taste buds on the papillae. But the bumps themselves are not the taste buds. They're just these things called papillae. The papillae come in 4 different types. The first 3 have taste buds on them, and then the 4th is an oddball that doesn't have taste buds on it. The biggest ones are the ones at the back of the tongue. There's about 12 of them, and those are called the valate papillae. Another name you might see in your lab guide or elsewhere, they sometimes call them circumvallate. You're welcome to use either name, I really don't care. And if you could take a microscope out, on each valate papillae, you'd find a couple hundred taste buds, okay? Scattered, so they're just towards the back of the tongue. Scattered all over the tongue, you have something called fungiform papillae. What does fungus mean, or fungiform? It means like mushroom shaped. I personally don't really see it, but someone thought that these little papillae look like little mushrooms. They're all over the place. They're a lot smaller than the valate papillae, so they only have about 5 taste buds on them each. Now, on the sides of your tongue, you may not have noticed this, but again, feel free to look in a mirror after class is done. On the sides of your tongue, you actually have stripes, okay? And those are called the foliate papillae. And the interesting thing about those is that when you're a kid, like a young kid, they have taste buds on them. But as you get older, you kind of lose the taste buds that are on the foliate papillae. And then the last type of papillae is the oddball, and it's also the smallest type of papillae, and that's called the filiform papillae. They're all over the tongue. They look like little cones. They kind of come to a point. They don't have any taste buds on them, but they do have tactile receptors. So they can tell you, they'll be connected up. They'll have, like, for example, Meissner's, Pacinian corpuscles on them, or they have, thermoreceptors, or they have nociceptors, but they don't have any taste buds and they don't have the chemoreceptors there, right? The real purpose of the filiform papillae, besides the tactile sensations, is it creates a rough surface on your tongue so that you can better, like, grip your food and manipulate your food when you're trying to, chew and swallow and all that stuff. Finally, the taste buds themselves are microscopic, but you'll have to identify this stuff in the microscope. So you're going to see some microscope slides of, like, foliate papillae, for example. And then if you focus in close enough on the foliate papillae, you will then see the individual taste buds. And this is what one taste bud kind of looks like under the microscope. It's sort of round, kind of looks like a little bit like an onion, I guess. And whoever discovered taste buds must have been similar or related, or kind of was friends with the person that discovered the olfactory epithelium, because you have essentially the same three types of cells. You got the receptor cell, which in this case is called the gustatory receptor cell, not the olfactory receptor cell. But then you also have, once again, supporting cells and basal cells, okay? The good news for you guys in lab is that you don't have to tell these three cell types apart in lab. You do for olfaction, you do for the olfactory epithelium, but for taste buds, you don't have to do that. Because honestly, it's kind of hard to do that. They all sort of run together, and it's really hard to point at one and say, oh, that's a gustatory receptor cell, or that's a support cell, or that's a basal cell. Okay. All right, so that's what the taste buds are going to look like. And then I'm going to skip ahead now to vision. And first of all, your eyes are able to see what's called visible light. Out in the environment, there's this thing called the electromagnetic spectrum that's made up of all these different types of light with different wavelengths and things. And you have stuff like gamma rays, X-rays, infrared rays, all this junk. But we can't see that stuff with our eyes. It's out there floating around, it's sending, I don't know, whatever, YouTube videos or funny cat videos, whatever. That's all floating around. You can't see it, right, but it's out there. The part that we can see is visible light, and it's just, you can see what they did on this diagram. They basically squeezed it into that little spot and then expanded it so that you could see. So we can see light that has a wavelength between 400 nanometers all the way up to 700 nanometers, which is a really small part of the electromagnetic spectrum, okay? And again, the way it's arranged, if you remember your colors from, like, art class, what was the mnemonic for the colors? Roy G. Biv, right? On this diagram, it's essentially sort of backwards there, but it would be like this, right? So, right, Roy G. Biv backwards. So you got your red. So up there you can see red has the widest wavelength at about 700 nanometers. And then as you go through the thing, all the way to violet has the shortest wavelength at about 400 nanometers. Now, in terms of the anatomy, we're gonna go over the anatomy of the eyeball in a second. But before we even get to the eyeball, we gotta talk about the accessory eye structures that kind of help out the eyeball. And that includes the eyebrows, the eyelashes, eyelids, the muscles that move your eye, the extrinsic eye muscles, and then the lacrimal apparatus. What does the lacrimal apparatus do?
Tears.
Makes your tears, right? The eyeball, your eyeball is about 2.5 centimeters in diameter. And you can see about 16% of the surface area when the eye is open. What percentage can you see when the eye's closed? I don't know. That was funny. No one liked that in the class this morning. They just gave me dumb looks like you idiot, just keep moving on. Very kind of similar to what you guys gave me. Yeah, actually. All right. Accessory eye structures. So eyebrows, eyelashes, they keep debris, the sweat out of your eyes, insects, whatever. They're there to try to keep junk out of your eyes. There's of course fancy names for all these things. The eyelids are called palpebrae. So you have your upper eyelid, which they call the superior palpebrae, and then your lower eyelid, which they call the inferior palpebrae. When your eyes are open, the space between the upper and the lower palpebrae, they call the palpebral fissure. If you take your eyelids and you look in the mirror and you pull down your eyelids and you look, you'll see that kind of shiny pink surface there. That's called the palpebral conjunctiva. So that's the mucous membrane that's on the inside of your eyelids. And you have it for your upper lids, you have it for your lower lids. You also have, and it has like little blood vessels in it. It's basically a mucous membrane. You also have a conjunctiva that's on the white part of your eye, the sclera. And that's called the bulbar conjunctiva. And that has little blood vessels in it. So like when, like if you stay up too late and your eyes get like bloodshot, they become red. Or you smoke the ganja, right? Sorry, dating myself.
The green.
What did you call it? The green? I don't even know what it's called anymore. What do you call it? The lettuce?
Lettuce.
How do you call it?
I don't call it that. Some people call it that. Like,
Some people, all right. Yeah, that's what I say. Yeah. I have some friends that use pot. Yeah, that's right. Anyway, that's little blood vessels that are in that bulbar conjunctiva that are becoming dilated to give you the red eyes, right? Of course, you can get an infection of both types of conjunctiva. And what's that problem called in layman's terms?
Pink eye.
That's pink eye, right? Or technically it's called like a viral conjunctivitis or a bacterial conjunctivitis. Another important thing to know, extrinsic eye muscles. These are the muscles that move the eye. One end of each, these muscles are kind of short. One end of each muscle is attached to the eyeball itself. The other end of the muscle is attached to the bones that make up the eye socket. So when these muscles contract, they'll pull the eyeball in different directions, right? These are very precisely controlled muscles, right? So there's lots of muscle spindles in them, because you need a lot of control. I told you when we did the cranial nerve chapter that each eye has 6 of these muscles, but I didn't give you the names of those muscles at that time, and now I'm going to do that. So there's 4 muscles. Well, there's 4 what are called rectus muscles. The rectus muscles are connected to, they attack like straight on to the top of the eye, the bottom of the eye, and then the medial and lateral sides. They're sort of connected straight on, if I could use that term. And then there's 2 more muscles that attach to the top and the bottom that kind of come in at an angle. They're not attached straight on, and those are called the oblique muscles. So there's a superior oblique, an inferior oblique, superior rectus, inferior rectus, medial rectus, and lateral rectus. If you remember, cranial nerve number 3 does 4 of those 6 muscles. Specifically, it does the superior rectus, inferior rectus, medial rectus, and inferior oblique. Cranial nerve number 4, the trochlear nerve, does the superior oblique, and then cranial nerve number 6, the abducens, does the lateral rectus, right? And it makes sense. The lateral rectus is on the sides of your eyes, so when it contracts, it pulls your eye away from the midline, which is the same thing as abduction, right? Abduction. Other muscles are important for opening and closing the eye. When you open the eye, it's due to the oculomotor nerve sending signals to this little muscle here, which you probably can't read from there, but that's called the levator palpebrae superioris muscle. That just means, palpebrae superioris means upper eyelid, and then levator just means to like lift it up. So when you open your eye, cranial nerve number 3 sends a signal to that muscle that basically just lifts your upper eyelid and you open your eye. When you close your eye, it's all about cranial nerve number 7 going to this muscle that you may or may not remember that goes around your eye kind of in a circle called the orbicularis oculi muscle. And when you send a signal to that, it'll contract and it'll close your eye. Now, I know what you're thinking. You're like, there's no way I'm going to ever remember these 2 things unless you show me a visual mnemonic that you've created that's 100 years old. Like, I got you. There. So there it is. This is dumb, but I've remembered this for like 30 years, and you're probably thinking it's stupid now too, but you'll remember it. So almost think of cranial nerve number 3 like pillars that are on a house, and put that cranial nerve number 3 in your eye to hold it open, right? You see where I'm going with this? And that's going to open up your eye. And then think of cranial nerve 7 almost like a hook, and you're grabbing your upper eyelid and you're pulling it down to close your eye. 7 closes the eye. Yeah? Now how dumb is this, right? All right. You don't have to answer. That was a rhetorical question. You don't have to answer that. Pretty fun. All right, lacrimal apparatus. This is what makes the tears. The lacrimal apparatus, or the lacrimal gland, is the part of the apparatus that actually makes the tears, and it's going to sit sort of superior and lateral to your eyeball. So it's sitting over like here, underneath the bone, of course, right? And you're going to have to. You should memorize. This is really dumb, but you should memorize the pathway of your tears. So the tears are made in the lacrimal gland. Then there's these little ducts that release the tears on the superior lateral part of your eye. And then the tears move across your eyes going medial and inferior. So they're coming across like this. And then in the medial part of your eye, you have these little openings that you've probably seen in the mirror if you look at your eyes closely, called the lacrimal puncta. Those are the openings that collect the tears. The lacrimal puncta are connected to these little things called the superior lacrimal canal and the inferior lacrimal canal. And they then dump it, if you're following over here, they then dump it into this area called the lacrimal sac. From there, the tears get dumped into another area called the nasolacrimal duct. And then finally, the ultimate destination of your tears is the inside of your nose, your nasal cavity. Okay? Tears are super important. They lubricate the surface of your eye. They have chemicals in them that can kill bacteria and viruses, so they protect against infections. You're making tears constantly. You don't just make tears when you're like upset and crying. You're making them all the time. And when you blink, every time you blink, you're spreading those tears across the surface of your eye to kind of keep it lubricated, etc. Okay. Now, even though the tears are going into your nose, you don't notice it because they're going in slow enough into your nose that your nose has a chance to basically just absorb it and you're not even aware of it, right? Obviously, if you're really getting your cry on and you're really upset, right? I think we talked about this before. What do you do when you're like really upset? You're like, and you keep snorting that stuff back up, right? Because you don't want to look like an idiot and be upset, right? So you're snorting that. That's because you're just making so many tears. You're kind of overwhelming the nose's ability to absorb it fast enough, right? Another thing that you might have noticed is when you have a cold and you've got like an infection going on inside your nose, that can block off the opening of that nasolacrimal duct, so the tears can't drain into your nose and they start to back up and they start to accumulate on the surface of your eye and you get watery eyes. Okay? All right, let me talk about the actual eyeball itself and then I think we'll finally be done today. The eyeball itself is made of 3 layers. The outermost layer is called the fibrous tunic. What does tunic mean?
Tuning something?
What's that?
Like tuning something?
Oh, like you're tuning something?
Yeah.
No, it's more like a clothing item, like a tunic is like a coat, okay? So this is called, that's like the fibrous coat, if you will, and it's called that because there's a lot of collagen fibers in it that make it very, very strong. You'll see, if, hopefully, we have the cow's eyes on Thursday, you'll see how tough it is to cut through the cow's eyeball with the scalpels, okay? Because there's a lot of collagen fibers in that outer layer, okay? So that's the outermost layer. The middle layer is called the vascular tunic. What does the word vascular mean?
No vein.
Just the opposite.
Lot of blood.
A lot of blood. So vascular means there's a lot of blood vessels in it. What's the word that means no blood? Not vascular, but yeah, avascular. You just gotta put the letter A before it. And then the retina is the innermost layer, right? And that's where all the hardcore physiology is. That's where the rods and cones are. That's what we're gonna talk about next week when we get into the physiology, right? Anyway, let's start from outside to in. The outer layer is the fibrous tunic. There's 2 parts to the outer layer. Most of it is the sclera. The white part of your eye is the sclera. It's got tons and tons of tough collagen fibers, okay? It makes it very strong. The word sclera literally means hard, okay? And it's really strong, okay? It gives the eye its shape, it helps protect the inner parts. You have sclera everywhere on the outer part of your eye except at the very front where it needs to be clear so that light can go into your eye, and that's where you have the cornea, okay? The cornea should be completely clear. The cornea is something that surgically they can reshape. If you have vision problems where you have to wear glasses because you can't see like distances or whatever, they can reshape the cornea using lasers, and that's called LASIK. Anyone in here had LASIK? No? People usually, I haven't either. I was like wussed out. I just didn't like the idea of it. But people love LASIK surgery.
It's actually bad. Like, so they say you're supposed to get it young,
Yeah.
but as you get older, eventually, like, there's like side effects. You get dried eyes.
I've heard that. I've also heard, your eyeball will still change its shape, so the surgery's good for a couple of years, but it could then, just stop working. Anyway, that used to be a very popular operation where they reshape the cornea, and what that does is by reshaping it, it will bend the light a little bit differently so that the image lands on your retina better. Yeah.
So is that why when I think of dogs can't really see where they get white in front of their eye? Is it covering the cornea? Or is
That may be cataracts that are building up inside of their lens. Cataracts are accumulations of abnormal proteins inside this part, and it
Okay.
can make the lens. It can make it look white on the inside. Yeah.
So this is the one like to see better, right?
To see better. Yeah. This is the one that to see better.
My mom got it done, but she says it didn't actually help. Like,
Really?
Not a lot. Yeah. 'Cause,
Okay.
she only got it done like in one eye.
Oh.
Like supposedly.
She needed to
didn't
do
need it,
two?
Like for the other eye, because like if she would have got it like done for both of the eyes, she wouldn't be able to like see from like far. Or something
Oh,
like
okay.
that.
Or she probably wouldn't be able to see like close if she
Yeah.
did both of them. Yeah.
But
Okay.
she says like it didn't really help. Like sometimes like she gets like blurry vision and like stuff like that. But her eyes were like red. And like
After
surgery,
after
like
procedure.
for a couple of weeks.
Yeah. I don't know. I was always too nervous to try it myself,
Yeah, I
but
wouldn't
like
do it.
a lot
I
of
wouldn't
people
Nah,
that have
it's
had
not-
it
it's
love
not
it.
worth it. 'Cause
She
it's-
had
it's-
big balls for that.
Cause it's really only it's really only a couple of years to see the benefits and like long
She's
term.
waiting for that.
Yeah, it scares me a little bit. Anyway, so that's the outer part of the eye. Now this, anyone know what the hell this is? Yes. It's a corneal abrasion. That's right. So this is a scratch on someone's cornea. Anyone in here have a scratch on their cornea?
Yeah.
It hurts like hell, right? when
My
you
buddy got a
scratch it.
bungee,
Oh, how did I do that? I
a
don't know.
bungee cord in the eye.
Oh, you got a bungee cord to
No,
the
a
eye?
buddy of mine. I got a bungee cord in the eyeball.
Ooh, okay. And he had a corneal abrasion
Yeah.
from it?
Is he blind
That was
now?
like a bad one.
Yeah,
It's
right
usually, so
And his whole
it hurts like hell,
That's the eye.
but most people do just fine afterwards. It's painful until it heals up. You might put a patient on some anesthetic eye drops to help with the pain. You might give them some antibiotic eye drops to just prevent infection. Bless you. But most people do fine with it. It's not usually a serious deal. It will heal up on its own. The scratches will go away. Why is it green though in this picture?
Yeah, it's
It's
really nice.
infected.
It's a dye that they put in, and then
Yeah, so if you see a patient in the ER or wherever and you suspect, based on their history, that they have a corneal abrasion, because the cornea is clear, you won't see anything if you just look at the cornea. Like if you take an ophthalmoscope and you look, you won't see anything because the cornea is clear. So you have to stain it in order to bring it out. And they have this dye called fluorescein. And the fluorescein's kind of interesting. The fluorescein itself is clear. It looks totally clear. You put a drop of this fluorescein on their eye, and then you take a special black light. You know, those ones that look purple, those black lights you could buy at, like, I don't know, wherever. And that black light will make that fluorescein fluoresce to this green color, and then you can see, like, the scratches that are there. Okay. What's the most common cause of a corneal abrasion? Anyone know?
Beach?
Would you say
The
beach?
beach?
No, it's actually just a finger to the
Yeah,
eye.
like right
All right,
here.
on accident. Yeah, I looks like it's Three Stooges or whatever, but just people. I presume like, I presume your own finger. I don't know, but yeah. All right. The second layer, the middle layer, is called the vascular tunic. The vascular tunic, also known as the uvea, it's called the vascular tunic because it has a good blood supply. By the way, the outer layer, the fibrous tunic, doesn't really have a good blood supply. It sort of depends on this layer underneath it to supply oxygen and glucose and things to it. The main part of the middle layer is called the choroid. It's pigmented. because it has a lot of melanocytes in it that make melanin. The purpose of the pigment is to absorb light when it comes into your eyes so that the light doesn't bounce around too much. If light bounces around too much inside your eyes, it can give you a blurry image. So the melanin absorbs the light as it's coming in so it doesn't bounce around too much. Okay. Another part of the vascular tunic is this little area here. That's referred to as the ciliary body. The ciliary body has two important parts to it, the ciliary process and the ciliary muscle. Ciliary process is what makes a fluid that's in the front of your eye called aqueous humor. We'll talk more about aqueous humor in an upcoming slide, and we'll talk more about it in class next week. The other important thing is the ciliary muscle. There's a little muscle that sits in that area that attaches to your lens. So this is your lens. The muscle is over there on the sides, and there's little ropes or strings that attach the muscle to the lens. And you need to be able to change the shape of your lens depending on how close or how far away the object is that you're looking at. If you're looking at an object that's far away, your lens needs to be really flat, kind of like this shape. But as you bring something closer and closer to your face, like if you're trying to read a book or something like that, the lens needs to change its shape and become more round, almost like a ball. And it does that by this muscle pulling on little strings that essentially change the shape of the lens. And we'll get more into how that works like next week, but it's just something to be aware of. The iris is the last part of the vascular tunic. That's the colored part of your eye. So whether you have brown eyes, blue eyes, green eyes, whatever, that's the iris. The iris is, it's really just a muscle, all right? And if you look close at your eye in the mirror, you can see the muscle fibers like running through the iris. And those muscles surround the hole in the middle, which is called the pupil. And by contracting certain muscles and relaxing other ones, you can either dilate or constrict your pupil, right? And again, which division of the autonomic nervous system is going to constrict your pupil? Parasympathetic. And which one's going to dilate it? Sympathetic. Okay? We'll get into that next week. By controlling the size of the pupil, you're controlling how much light gets through and ultimately goes to the back of the retina where all the kind of hardcore physiology is taking place. The color for your eye is due to melanin once again. And it's the same melanin that gives you your skin color, all right? There were two, if you remember from the skin chapter, there's eumelanin and then there's pheomelanin. If you have, darker skin or brown eyes or whatever, you probably have more of that eumelanin going on. If you have fair skin or blue eyes, you have more of the pheomelanin, and if you have like green eyes, it's like a blend of both or something like that, right? This is a picture. You'll eventually, make sure at some point you spend some quality time with pictures like this. We'll go through this stuff in lab on Thursday, but that's just kind of showing you all the parts. And then lastly, the innermost layer is the retina. The retina is where all the hardcore physiology takes place that we'll cover next week. There's two parts to the retina. This layer here is called the pigmented retina. It's kind of like the choroid was. It's got melanin in it that can absorb light so that it doesn't bounce around too much. But the part we're going to focus mostly on is this part called the neural layer, which is all the rest of this down here. And the thing that's going to be tricky for you on Thursday in lab is you're going to see microscope slides that look like that of the retina. And there's different layers that you can see listed here. And you're going to have to identify all those layers. And when you first look at it, it just kind of looks like a bunch of stripes. And you're like, what the hell am I looking at, right? But I'll eventually I'll show you how you can tell these different layers apart so that you get it right on the practical. If you go into the exact center position of the retina, in other words, if you could take like a ruler and just draw a straight line right through the front of your eyeball and follow it all the way straight to the back, you would hit a spot on the retina that's called the macula lutea. It literally means yellow spot in like Latin, that's the translation. And in the middle of that yellow spot is an area you may have heard of called the fovea. The fovea is an important area because it has a lot of cones in it, and it gives you your sharp color vision. Anytime you look at an object, or you look at a person, you look at their face or whatever, you will use your eye muscles to try to turn your eye so that the image comes through and hits that fovea. When you look at things, when you really focus on things, you're always trying to move your eyes so that the image lands on the fovea part, because that'll give you really sharp vision, okay? This is a view of someone's retina using an ophthalmoscope, and you can look for like different things that are on there. You can look for dilated blood vessels that might indicate diabetes or high blood pressure, things like that. The optic disc is something you can see on the eyeball. The optic disc usually sits just to the side of the fovea. So like if you trace this through like this, straight through, the fovea would be right about there. And then to the side of it, this area would be the optic disc. The optic disc is an important spot because it represents where the optic nerve attaches to the back of the eyeball. So if you could follow straight through from the optic disc, that would be where the optic nerve is, cranial nerve number 2. One thing that's special about the optic disc is there's actually no rods and cones there. So it's a physiologic blind spot. That means that when you look at something, if an image comes through your eye and it lands on where that optic disc is, it's like that object isn't even there. And there's a fun activity, I think it's still in the current edition of the textbook, it's not that much fun, but it's fun for me. But you take your textbook and there's a picture in there where it's got 2 dots, and you're supposed to cover one of your eyes, and then with the eye that you're looking with, you're supposed to focus on one of those dots and just keep the eye on that one dot. When you get the book at a certain distance from your face, it'll be like that other dot that was on there magically disappears, and that's because that image is hitting the blind spot. I spent one weekend just doing that, and it was a lot of fun. You just keep like this, and it just keeps disappearing, right? All right, rods and cones, we'll talk more about these guys later. These are the guys, these are the photoreceptors. Rods do black and white images only. Cones are what give you the color vision. Rods are good in low light conditions. So as it starts getting nighttime out, you lean more into your rods because they're more sensitive. But when there's good light conditions, you lean into your cones because they give you color vision. Lastly, last slide, cavities that are inside your eyes. In the front of your eye is an anterior cavity. Notice, so the cavities are kind of based on the position of the lens. If you're to the side or in front of the lens, you're in what's called the anterior cavity. If you're posterior to the lens back here, you're in the posterior cavity, also called the vitreous chamber. There's going to be fluid in both cavities. The anterior cavity is going to have what's called aqueous humor in it, which is very watery. The vitreous chamber is going to have vitreous fluid in it, which is more like a jelly. It's like a clear jelly that sits back there. All right, that's all I want to say. I know it was a lot. Thanks for sticking with me. If you have any questions, don't forget about that. Make sure you're here right at 8 a.m. because we'll do the practical first thing. I'm not 8, sorry, I apologize.
What's going on?
Sorry, I'm thinking of my class this morning. 11 a.m., make sure you're here right at 11 a.m. We'll do the practical on Tuesday. All right, so we already did kind of the anatomy of the nose, we did the anatomy of the tongue, we did the anatomy of the eye, now we're going to do the anatomy of the ear, so I'm going to jump all the way ahead to number 43, and that's where hearing starts, somewhere. Here it is. All right, so hearing is the ability to perceive sounds, doi, right? The ear is an art engineering marvel because it can censor. I didn't write this sentence. Can reduce sound vibrations and amplitudes. I have no idea how big a diameter of an atom of gold is. I guess this is impressive, right? An electrical signal is 1,000. Okay, whatever. The ear also contains receptors for equilibrium. So don't forget that. Everybody thinks of the ear as being hearing, but it also does equilibrium. Equilibrium means kind of knowing where your whole body is in space. Like, your muscle spindles, your Golgi tendon organs, those things can tell you, like, whether your arms are bent, whether your arms are straight. They can tell you whether your arms or legs are moving. But those can't tell you the position of your entire body, for example. Like, I could be standing like this, right? I'm standing like this. I close my eyes. I know that my arms are bent. I know my legs are bent a little bit. I know all that stuff from the muscle spindles, the Golgi tendon organs. But what I don't know is my entire body right side up like it is, or could I be holding this exact same position, but just my whole body be upside down? The Golgi tendon organs, muscle spindles, they can't tell you that. That comes to your inner ear, telling you kind of the position of your body relative to gravity. Are you upside down, right side up? Are you tilted a little bit to the left, a little bit to the right? Your inner ear can tell you that. Your inner ear can also tell you, like, you know, is your body accelerating or spinning? Like, you know, I could be just standing steady like this, or maybe like I'm a figure skater and I'm spinning in a circle. Like, how do I know it's not coming from the peripheral proprioceptors, like the muscle spindles, Golgi tendon? It's coming from your inner ear, okay? Anyway, so in terms of the anatomy, there's three principal regions to the ear. The external ear, or the outer ear, is basically a funnel that draws the sound waves in. Then you follow this kind of tube that eventually you reach your eardrum, and then on the other side of the eardrum, you're in the second part, which is the middle ear, which is basically a cavity, and that's where you're going to have your three ear bones that we'll discuss them again, but you already just learned about them in the exoskeleton chapter. And then on the other side of the middle ear, then you come to the internal ear. The internal ear has a bunch of weird sort of twists and turns and passageways. It looks like a maze. Excuse me, so it's actually sometimes referred to as the labyrinth because it looks like a maze. The internal ear is filled with a lot of fluid. So not to get too much into the physiology, but what's going to happen is sound wave comes into your ear, it moves the eardrum. When the eardrum moves, it makes the bones move. When the bones move, it causes a fluid wave in your internal ear that will get converted into an electrical signal that you'll perceive as sound. Okay, that's like the big picture of the physiology. So let's talk about the external ear first. The big floppy part, the part that's your ear, like people say your ear, they're talking about the auricle, or it's also known as the pinna. It's basically just skin overlying some elastic cartilage. What was the claim to fame for elastic cartilage? It was the whattest cartilage in your body?
Brings you back to
I think you're saying it.
Most elastic.
Yeah, it's the most it's the most flexible. Yeah, flexible, elastic, whatever you want to call it, right? Remember, hyaline cartilage was the most common, and fibrocartilage was the strongest. Elastic cartilage is the most flexible. If after you are at the auricle, you then have this long passageway that you're not supposed to put things in, called the external auditory canal. That goes by a couple of different names for the lab practical next week. You can call it the external auditory canal if you want to. They sometimes call it the external acoustic canal, that's fine. They also call it the external auditory meatus or the external acoustic meatus. Any one of those names is fine, I don't really care. You eventually come to an end when you reach your eardrum. The fancy name for your eardrum is the tympanic membrane. And that's the divider between your external ear and then your middle ear. Along the way, lining that external auditory canal, you'll have the openings of various earwax glands, which are called ceruminous glands. They're basically just like modified sweat glands that mix sweat with some oil, and it gives you like the earwax. Now, once you're inside the middle ear is basically a cavity that's been cut out of your temporal bone. And inside you're going to have your three ear bones that you already learned about. The fancy name for the ear bones is the auditory ossicles. And so if you look at this, attached to the inside of the eardrum is the malleus, or the hammer. The malleus forms a joint with the incus, also known as the anvil. And then the incus forms a joint with the stapes, also known as the stroke. Now, the stapes overlies a little opening in your inner ear called the oval window. So the flat part of the stapes is sitting over this thing called the oval window. So again, not to get too much into the physiology, but a sound wave is going to come in through your auricle. It's going to the sound wave is going to pass through the external auditory canal. It's going to make your eardrum move. When the eardrum moves, the malleus moves. When the malleus moves, the incus moves. When the incus moves, the stapes moves. And when the stapes moves, it pushes against the internal ear and it creates a fluid wave inside of your internal ear that will eventually get converted into an electrical signal that you perceive as sound. Okay. Besides the ear bones, something else you should know is you also have your two smallest skeletal muscles in your body in your middle ear. So you got the two smallest bones, or you got the three smallest bones, and then you also have the two smallest muscles, and they're called the tensor tympani and the stapedius muscle. And these little muscles are attached to the ear bones, and they're designed to reduce the movement of those ear bones with loud sounds. Okay? In theory, you know, the louder the sound, the more the eardrums will, or the more the bones will be moving, and they could potentially move so much that over time they would start to damage themselves. So to prevent too much movement from happening and kind of reduce the impact, these little muscles will contract. These will help you with prolonged loud sounds. So like if you're going to like a concert or something that's loud, they would probably help with that. You know, they'd start to contract after a while to reduce the amount of movement of the ear bones. They're not going to help with sudden long sounds, right? So if somebody sneaks up behind you with like a cymbal and goes tsh, like that in your ear, they're not going to have enough time to contract to really prevent the ear bones from moving.
So is that what the ringing of the ear effect is, basically?
No, the ringing of the ear effect usually comes from the internal ear and stimulating the cells that are in there, kind of causing the cells to be permanently turned on, even when there's not a fluid wave going, causing them to still be stimulated. So it's something deeper in. Yeah. Another thing to know about the middle ear is that this is where your eustachian tube is connected, okay? Which I think a lot of you guys have probably heard about before. Eustachian tube, also known as the auditory tube, connects your middle ear with your throat, okay? Specifically, your throat, or your pharynx, has three parts to it. The highest part of your throat, which sits right behind your nasal cavity, is called the nasopharynx. And this tube will connect the nasopharynx to the middle ear. And in a child, this tube, because the entire skull of a child is relatively small, and because of the shape of the child's skull, this tube is relatively short and it's relatively open in the child, which is why in children, anybody that has young children knows that if you get a throat infection in a young kid, what usually follows?
Ear infection.
An ear infection, all right? Whereas as your skull grows, this passageway becomes longer and it tends to collapse more, so that in an adult, it's a lot harder for little bacteria that are in your throat to be able to climb all their way up through this tube and get into your middle ear. So you don't tend to see ear infections as often once you become an adult, just because your whole skull has basically changed. Okay. The purpose of having this, this tube is to equalize pressures on both sides of the tympanic membrane so that it doesn't get too sucked in or too pushed out, to kind of keep it in a centralized position. So, whoops. So if you think about it, you've got air from the outside world coming in through your ear this way, right? So air from the outside world is coming in through that way. But then you also have air from the outside world coming in through your nose and then going up through this tube on this side of it. So by having air on both sides of it, it kind of equalizes the pressure so that this remains in sort of a neutral position and doesn't get pulled in or pushed out or whatever. Now, obviously, if you like go to like a high altitude or something, you've been flying or whatever, and your ears pop or whatnot, what's happening is the pressure from the outside is kind of pushing this eardrum in, okay? And because when you get older, this tube is sometimes collapsed, in order to solve that problem, what can you do? You could chew gum or swallow really hard, and that should force air up through that auditory tube onto this side of things, which will then kind of push this back out again and equalize the pressure on both sides. Okay? This also leads me to another extra credit opportunity. So get your pencils, you might want to try this now. Actually, pencil is one of the things you're going to need. So you'll need three things for this extra credit opportunity. You'll need a sharp pencil, you'll need a bucket of water, and then you'll need your ears. Okay, that's all you'll need. And here's what I want you to do. I want you to take the sharp pencil and I want you to push it into each ear, and I want you to put a hole in the eardrum. Okay, that's step one. This is the first step. Put a hole in each of your eardrum, and then I want you to take that bucket, and I want you to put your face, including your nose and your mouth, in that bucket, and then I want to see if you can breathe through your ears. Okay? You should be able to do it, right? If you think about it, if there's a hole there in there, it should be able to go all the way down and get into your lungs. You're up for it, Dan?
We gotta record it?
Yeah, you do have to record it. Yeah, please record it. How much? All right, don't do that. It was a joke. I know some of you are desperate, but please don't do that. All right, it was a
If you
joke.
don't give extra credit, that's the only.
Yeah, no extra credit, nothing. Don't do it. But in theory, yeah, you should be able to breathe through your ears. All right. Last but not least is the inner part of your ear. The inner part of your ear, the internal ear. Like I said, they sometimes call it the labyrinth because it looks like a maze. There's a lot of twists and turns and passageways, and they're all filled up with fluid that we'll talk about. And basically, if you can view the internal ear as being kind of carved out of your temporal bone, all these twists and turns that are carved out, and they refer to that kind of carved out cavity in the bone as the bony labyrinth, okay? And there's three parts to the bony labyrinth: the semicircular canals, the vestibule, and then the cochlea. Inside of the bony labyrinth, inside of the cavity part, are a series of structures that are referred to as the membranous labyrinth, okay? So for example, inside of the semicircular canals, which are the bony labyrinth, are something called the semicircular ducts. Inside of the vestibule is membranous labyrinth that's called the utricle and the saccule. Inside of the cochlea is a membranous labyrinth called the cochlear duct. All right, so the bony part is just the cavity, and then the membranous labyrinth are these things that are inside of it. Here's the picture down here. So shown like over here, like in light tan, that would be a bony labyrinth over here. Shown here kind of in brown, that's bony labyrinth there. And then shown in red over here and in pink in there, that's the membranous labyrinth that's inside of the bony labyrinth, if you will. All right. So let's talk about these three areas then. So the middle area, the bony labyrinth in the middle area, that's called the vestibule. The membranous labyrinth that's inside the vestibule is this thing called the utricle and this thing called the saccule. All right. What the hell does this stuff do? This stuff does static equilibrium. There are sensory receptors there for static equilibrium. That's the type of equilibrium that tells you where your body is relative to gravity. Like, is your body upside down? Is it right side up? Is your entire body tilted to the left or to the right? Okay? Muscle spindles, Golgi tendon organs, joint kinesthetic receptors, they can't tell you that kind of information. They can tell you if your arm is bent or your leg is straight or if your knee is moving, but they can't tell you, are you right side up, upside down, tilted to the left, tilted to the right, all right? That's static equilibrium. We'll talk about how static equilibrium works next Tuesday. On one side of the vestibule area, you have the bony labyrinth called the cochlea. Inside the cochlea I mentioned is the thing in red, that's the cochlear duct. And as soon as you hear cochlea, you should think hearing, okay? That's where you do your sense of hearing. And then the other part of the bony labyrinth is called the semicircular canals. Inside of the semicircular canals is the membranous labyrinth, which is referred to as the semicircular ducts. This is also going to do equilibrium, just like this guy did equilibrium. This guy does equilibrium, but this is a different type of, this does what's called dynamic equilibrium. This is equilibrium that will tell you if your entire body is moving or not. Not whether like your joints are moving, okay, that's joint kinesthetic receptors, but like, is your entire body accelerating? Like if you're a passenger in a car and you close your eyes, you can feel whether the car is accelerating, decelerating, things like that. Or if you're like an ice skater, you can tell if you're holding this position versus are you just holding this position or are you actually holding this position but you're spinning around, right? You can tell that kind of movement information from dynamic equilibrium, and that's handled by these semicircular canals. Now, I wanted to show you this slide. So this is a picture of kind of what you're going to see in lab today under the microscope when you look at the cochlea. Okay, this is basically a diagram of a cut through one turn of the cochlea. The cochlea, by the way, does anyone know what the word cochlea means? Not shell, but close. Means snail. All right, kind of looks like a snail with its weird sort of, thing there. Anyway, what they've done in that next picture, they've taken, notice how it curls around itself several times? Well, what would it look like if you just took a cut through one of those turns? And the answer is, it would look like this. This is a close-up of a cut through one of those turns. And you're gonna have to look at this under the microscope and find these different parts. There are 3 sort of cavities to look for. The membranous labyrinth is in the middle, and that's referred to as the cochlear duct, shown in kind of red there, or in this diagram, the red thing in the middle, that's the cochlear duct. On the sides of it are the other 2 cavities that are listed here. One of them's called the scala vestibuli, the other one's called the scala tympani. All 3 of those cavities have some type of a liquid inside of them. And when you hear something, the bones in the middle ear will create a fluid wave in these cavities that will eventually get converted into electrical signal. Okay. So some tips when you look under the microscope today, try to find these 3 structures. They'll always be sitting next to each other. The one that's in the middle will always be the cochlear duct. And then on one side of the cochlear duct will be the scala vestibuli. On the other side of it will be the scala tympani. The way to tell which is which is try to find this vestibular membrane. It's a really thin, it almost looks just like a little rope. It's a really thin membrane. And if you can find that, that'll tell you the side where the scala vestibuli is. If you find that thin rope, one side of it in the middle is going to be the cochlear duct. On the other side of it will be the scala vestibuli. On the other side of the cochlear duct, there's another membrane that's a lot thicker called the basilar membrane. And sitting on top of the basilar membrane is this thing called the spiral organ or the organa cordi. And that's really where all the hardcore physiology takes place. That's where the physical fluid waves are actually converted into an electrical signal, which we'll get into those details next week. All right, and then I wanted to skip a couple of slides and just show you another picture that'll be helpful in lab today. This is a picture very similar to what you'll see in lab today. This is showing you an area of the semicircular ducts that it's referred to as the ampulla. So again, here's a big picture of it. You can see there's the semicircular ducts. At the base of each duct is kind of a dilated area that they call the ampulla. Or if I go back to this picture, the ampulla would be like this spot here that's slightly dilated, this spot here, or that spot there. That's the ampulla. And if you look inside the ampulla, you're going to see this thing that looks like a little hill. The name of that little hill is the crista. Sitting on that little hill are special sensory receptor cells called hair cells. There's a lot of hair cells, and there's a lot of talk about hairs. When you talk about like olfaction, there's olfactory hairs, or gustation, there's gustatory hairs, or here you have hair cells, or in the cochlea you have hair cells. None of these things are actual hairs, just so you're aware of that. All of these, when they say hair, what they really mean is like cilia or microvilli. They're not, I don't know, they just thought they looked like hairs, so that's why they use it. They're not like the hairs on your head. I don't want you to think that. But anyway, sitting on this little hill are these hair cells, and connected to the hair part of the hair cell is this jelly-like thing called the cupula. And I don't want to get into the physiology, but what's going to happen is when your body spins or moves in one direction or another, there's fluid inside of all of these semicircular ducts. And the fluid, as you move, will move across this cupula and cause it to move to either the left or the right. When the cupula moves, it's going to yank on the hairs of those hair cells, which are not really hairs, but it's going to yank on those hairs, and that's going to be converted into an electrical signal that you'll perceive like, oh, my body's spinning to the right, or oh, my body's spinning to the left, or whatever, okay? that's just a little bit of the physiology of what's going to be, what's happening, in case you're wondering. Inside each of these hills, connected to the hair cells, is a nerve that'll take the signal back into your brain, and that's called the ampullary nerve. And you can see it there. Okay? So that's it. That's all I want to talk about in lecture. Now, for the rest of the time, I want you to do lab stuff. So there are several microscope slides to look at. There's several models to look at. Let me just tell you, for the microscope slides, there's a whole box back there. Some of the stuff in the box you've already looked at. You don't need to look at it again. Like some of it's like skin and scalp and stuff like that. But the 5 slides back there that you should look at are these ones that are listed here. So olfactory epithelium you should look at. That's from the nose. Foliate papillae, that's from the tongue. Retinas from the eye, and then these 2 are both from the ear, the cochlea and the crista ampullaris. So look at those 5 slides, and then there's also a bunch of eye models back there, and there's a bunch of ear models and tongue models. Look at all that stuff. And then in a little bit, I'll give you a little bit of time to look at that stuff. I will have, I guess, 3 people, whoever wants to do it, can actually dissect the cow's eyeball, and I'll show you how to do that.
I got coke.